let me just make sure that that's going on all right.
It's recording already.
Very good. And now I'd like to go ahead and just introduce Caitlin. So Dr. Caitlin Stack-Whitney is an insect ecologist and assistant professor in the STS Environmental Studies program at the Rochester Institute of Technology. She studies insects, ecosystem services, and landscape context in overlooked human-made habitats like roadsides. She has previously developed and published several open educational resources, including one using NEON data, and participates in the EREN Got Milkweed project in one of her classes. This new EREN-NEON Flexible Project module on backyard pollinators builds off these experiences and interests to invite students to explore closely wherever they are. So I will now turn it over to Caitlin, and we'll go from there.
Well, hi, I'm excited to talk with everybody and hopefully collaborate all together the semester ahead. So I'm going to try to share my screen. So patience with me for 1 second. Hopefully. Can someone give me a thumbs up if you can see my screen now? Excellent. And so if you're listening on the phone too, these slides are available so we can make sure you can see them later. So just one note that my apologies if the orange is a little bright. I use this particular template because it gives space for the captioning at the bottom. So if you weren't aware, there's closed captioning available. You might have to hit show subtitles. So if you want the captions on for this meeting, that's enabled in Zoom, and there's a remote captioner captioning this meeting right now. And thank you, Lori and Tim, for the introduction and for shepherding us all on this exciting new journey for the semester ahead. And so today we want to talk about, we're calling it backyard pollinators, but no one needs a lab to do this. And so this is a project where we're going to have students observing insect pollinators in backyards or nearby natural areas across urban-rural land use gradient to look at interaction networks of plants and pollinators. And so why do we want to do this in the first place? What's the motivation? So pollinators are critical. This particular graph is showing, in particular for insect animal pollinators, the dependence of agricultural production on pollination by animals as current as 2012. And you can see that in the US, for example, it's under 10%. That's an enormous amount, though, of crop production that's relying on insect animal pollination. Pollinators are also really diverse. So I'm biased, I'm an insect ecologist, and this is in particular going to be focused on insect pollinators. But one of the things we can do is explain to students that there's many, many different kinds of pollinators, including mammals and bats and birds, and even, you know, for example, one of the other EREN-NEON projects is going to be on mosquitoes. Mosquitoes can also be pollinators. So lots of organisms that can be loved or not so loved can be pollinating and critical for ecosystem services that we want. But also we know that pollinators and pollinator habitat is declining. And so this figure here is looking at a decline of pollinating insects from a paper that was published in Nature Communications in Britain in particular. But one of the things to keep in mind is that looking at abundance of an organism is not the same. Co-occurrence, right, of a plant, for example, an insect, is not an interaction. And so this lab activity in particular is actually using direct observation of plants and pollinators together to look at interaction networks. So I think that's something that's also really neat we can talk to students about, is that there's a lot of concern about insect declines, and we're looking really critically about how we can assess that question. And so this is in particular not just looking at, is an organism present, but what is it actually doing? So we're going to be using a standardized technique. Sometimes it's called timed focal floral observations or flower visitations to actually get a proxy for pollination, not just the presence of pollinators. And we want to do this using really simple materials. My semester, for example, starts next week, and I'm supposed to be teaching in person in a socially distant way, but many of you may be teaching people who are at home. So the goal is to do this with materials and with methods that we can scale collectively, where we're getting cool ecological insights, hopefully doing a neat collaborative project together, but also in a way that's really possible for our students and our classes that are operating in a really different variety of contexts. Okay, and one of the things I want to point out is that we're going to be focused on beetles in particular. So next thing I want to explain is why am I so interested in us looking and trying to find beetle pollinators in particular. And so one of which is that beetles are generally understudied and undervalued. So there's been some research that's shown that people tend to focus on bees or really large pollinators, and we're actually don't have as much information in part on other kinds of non-bee pollinating insects like beetles. One of the major reasons for that is that beetles are not crop pollinators. So they're actually known to visit and pollinate dozens of native plants, but none of them are crops. And I'll show you some pictures. I've been going around looking for beetle pollinators to show you in my own backyard and on my own campus this past week. And so I did find some. So it's definitely possible if you're thinking of doing this lab in the coming weeks in a temperate region that you'll be able to find beetles on flowers. So that's really exciting. And I think it's a neat way to, take one of the most diverse taxa on the planet and show that they're also participating in this critical ecological service, even if it's not for crop production. And one of the reasons that they're undervalued is they're getting less attention. This is just a screenshot of an article I wrote for American Scientist about the federal pollinator plan. The federal pollinator plan was passed under the last administration, and it actually only focuses on 2 organisms, monarch butterflies and honeybees. So you can see no mention of beetles anywhere in our national pollinator plan. And so this is a good opportunity to remind students that there's lots of other insect pollinators out there, even if they're not getting that attention on a policy or a scientific level at this point. Okay, the more immediate inspiration, one of the reasons I approached EREN about this was 2 things in particular. So one is that we're living in a rapidly changing world, not just climate, but also land use. Urbanization is happening rapidly around the planet. And this is a figure from a paper that was published in 2013 that looked at plant pollinator interactions and seemed to find, this was one study based around one city in France, that more urban environments had fewer interactions between plants and pollinating insects. So there's concern that as the planet is changing, we might actually be losing these interactions, which remember is different than just saying, do we see insects? We might actually be decoupling these interactions, right? Which could have catastrophic effects in terms of thinking about pollination, not just of our food, but all kinds of other flowers. The more immediate, also one of the inspirations was a lockdown garden survey that was led by Dr. Jeff Allerton. Now, I do want to point out that this was really cool. This is one of the graphs they made of their interaction network across the EU this spring. This is at a species level. And so I want to point out a couple of things that will be very different between what I'm talking about and this plot. One of which was they only did this in the spring. It was expert taxonomists who were able to identify everything to species themselves with no help. So I would not expect that of students, and we're not trying to get there. We're going to be using functional morphospecies groupings for both plants and insects. So this might be something that experts can do with each other as a hobby, but we're not trying to replicate that. And we have some cool hypotheses that we want to test on the way. But I think one of the things that's really exciting about this is that they can compare their results to this survey in the EU. And this one was done under lockdown, literally with backyard specimens, people's backyard gardens. I think it's a great reminder for students that we can do cool science no matter where we are, even under quarantine conditions, right? So we're in a rapidly changing situation, and this is truly science that we can do in a variety of settings, including in container gardens. All right, so this project in particular, what is it going to be about? What's our objective? So I mentioned before that Geslin paper, there is some limited research that shows that these interaction networks can be reduced in more urban settings, but those were actually using sentinel plants. And a lot of that other research was done using crops in particular. So the big objectives are, I think, twofold of this. One of which is that I hope collectively, all of us on this call and anyone else, right, that we want to do the largest non-crop and non-sentinel plant pollinator interaction study ever done. So I mentioned to you that Geslin study was just done in one city. We have the opportunity to do a really large scale observational study that's outside of cropping systems and look at who's showing up where and actually learn a lot at a continental scale potentially. And the other is something that's safe and close and fun, where we're just helping students pay close attention to the world around them in a way they maybe didn't do before. And so I'll talk more about this picture on the right, but this is a quadra that I brought with me last week to my own campus. So that's kind of where we're going. That's what's guiding this particular lab. And we have 3 hypotheses that we can test here based on previous literature. And so one of which is that as urbanization increases, I expect that the complexity of the network, literally the nodes of interaction that we're going to see in terms of students doing this individually or compiling our class data, is that we're going to see the complexity go down. And that's in part informed by previous research. The next hypothesis we can test is that we will see fewer floral visitation events in more urban landscapes. Not just the interaction network is less complex, but that overall we actually see fewer insects visiting flowers in a more urban landscape. And the third is that in particular, I expect to see fewer beetle floral visitation events in more urban landscapes. And that's again informed by some previous research, including that beetles can be a sentinel species for urbanization in particular. So we have a specific hypothesis in particular for beetles. And then later I'll point out to you, we have another hypothesis that's also specific to beetles. So I'm gonna walk through first this core module that we have, and this is where the one where we really want to pool our data together. And then there's all these add-ons. So basically, if you want to do this as a 1-week lab, it's set up so that the core is something you could do across, for example, 1 to 2 class periods with some background reading and a training exercise and then carrying it out. But if you want to expand this across more time in weeks and incorporate other elements, it's very scalable to add on other dimensions. And so because I'm expecting that everyone I'm talking to right now is the instructor, we're going to be basically in the weeds of how you would carry this out and all the elements that this module will be providing and all the add-ons would provide. Okay, so the core module itself, we have 6 learning goals for this particular core module, and I'll just walk through them quickly. And it's mostly to be able to explain later when we talk about all the other add-ons, what else you might get from adding those other ones on if you want to. So we want students to develop hypotheses about organisms, communities, and ecosystem function. We want them to actually identify plant and pollinator functional groups based on guides that will be provided to them to this exercise, and to record, enter, and organize both qualitative and quantitative data about those plants and insects and the site where they're doing this observation. We also want them to analyze their data and collaboratively, hopefully their class data. As we get data in from each other's classes, we'll be able to also keep sharing the data from the project itself. So the goal is not for this to end this semester, but to keep doing it potentially every year. Now, fall, here at least in upstate New York where I am, right, there's plenty of flowers still out. This is very possible to do, but it'll be unlikely to be possible to do this coming spring. It's generally still winter for many of us in the north. If you're in the southeast, it might be possible to do next semester too, but we can keep growing this data set to share with each other every year to even use in class and hopefully do some other things with. So we want to be able to compare results between sites and evaluate patterns, and then talk about the importance of this standardized technique that we'd all be doing, but the value that comes from doing this at scale together over time and space. So what are the components if you chose to adopt to do this lab activity in your class? So there would be a background PowerPoint/reading. So you can decide what format would make sense for you. It'd be the same information, and I will also record myself giving the PowerPoint. So your choice would be that you could give the PowerPoint, that you could assign me giving the PowerPoint that's recorded in advance and captioned, or they could read it as a lab packet, right? So depending on what format your class is in, that's essentially an hour of material that's explaining what we're doing and why. And you could decide, do you want to give that information? Now, depending on what course you're fitting it into, if it's intro or bio or ecology versus if it's a different kind of class that's more focused on insects, there might be things that you want to cut out or add, right, based on your own course needs. So that's one of the reasons to potentially use that PowerPoint and slice and dice it as you choose instead of using a pre-recorded video. Based on feedback from Lori and Tim talking with some educators, I believe it was last week or the week before, there was some feedback that they wanted the opportunity to do a training and give students a quiz. So that's one of the things that we'll be building into this, rather than just saying, here's why we're doing this lab activity, let's go do the lab activity. They wanted students to have an opportunity to actually try it out in a essentially zero stakes setting online, and to be able to retake that and do it as a training exercise. So that was a great thing to get feedback about, to say, okay, we can provide that, not just a guide itself for doing the activity, but actually a training quiz to do in advance, potentially as a homework assignment before the activity itself. There will be a template for data entry and for compilation, some templates for data analysis, and then questions for reflection for students. So those are all the pieces of the core module if you chose to adopt it. Okay, so if you want to do this core module, right, what else was incorporated is from NEON in particular. This core module uses NEON plant cover data and some methods, and also talks about the importance of phenology and explains how NEON phenology data is incorporated. And those will mostly be incorporated into the background PowerPoint. We would not ourselves be using the exact methods that NEON uses to do this lab. But so I'll be explaining to you for each of the add-ons what elements of NEON data are incorporated, or which data sets. Okay. So what materials do you need? Now, the goal of doing a focal floral observation is truly just to look at a focused set of flowers and observe what insects land on here. And in order to standardize this across everyone, we look at a particular size area. So this is a 50 centimeter by 50 centimeter square. And we look at it for 10 minutes. So on one hand, it's a very short lab, but it's a focused observation for 10 minutes. And we also need to know some information about the site. So it will not take students 10 minutes, right? This, I think, would be a full lab activity. The other thing that I want to point out is this is my PVC quadrat. If you are on a campus or you have a lab budget and you're doing socially distanced labs like I'm doing in person, you can make cheap PVC quadrats, but you do not need to. So you can do this with other materials. I was just telling Lori before this that I figured out how many pieces of paper you need to rip up to do this with printer paper. So you can set up quadrats in a lot of ways. The goal is to make sure that you're looking at the same size space. So there's lots of ways that we can do this. One of which I just set up in my backyard was using toothpicks and twine. So I took a measuring tape and I measured out 50 centimeters. And then I measured out my twine, and then I actually staked it using toothpicks in my yard to actually do a 10-minute observation. So that worked perfectly fine also. And so I only use these 4 materials so that the really critical pieces for the observation are the training. It's not so much that you need specialized equipment, but they need special information to really carry out this lab successfully. All right, so my apologies that my twine is green, but there's a picture right there of my twine quadrat, if you can see that, but unfortunately on my yard it's a little bit tricky to see. So that's that was my twine quadrat. So one option to consider is if you're in a position to mail out materials, or if you were planning on printing a lab packet for students and didn't have quadrats, the picture you're looking at here is actually 4 toothpicks and a piece of string that's already been tied in a knot to be the size of the square. So if you're in a position to give out those materials to students, that's one very cheap way to make sure that they have the materials, to just literally give them the toothpicks and the string. Okay, so that's what they would need if they're going to head out, but what do they need to do? So these are just some pictures from my own campus personally that I'll just show you that if we're looking at a picture like this of a quad, you might be thinking, well, I don't really see great pollinator habitat there. Where will my students do this lab activity? How could they do this here, right? But 50 centimeters by 50 centimeters is actually really not that big. So in this picture here, they can go to those flowers that are in the foreground. It actually takes up an entire quadrat. So just to get a sense of scale, this is not very large. So here's another picture of one of the quads on my campus, and it has this little green space in the middle of a walkway. And we were able to also do the observation actually in even that really small patch. And often students are really surprised that even in these patches that seem to be in a sea of grass, that insects will show up onto these flowers, especially if you're sitting for 10 minutes. One tip I would give is if students are not able to place something like a quadrat or string very quickly, like if they were trying to, arrange paper, is to leave a minute between setting up your, complicated quadrat that you're MacGyvering together and when you actually want to start the observation, because you might have disturbed everything in the area and they all might have flown away. Okay. One thing to keep in mind, encouraging students to be creative, this is a picture of my front lawn, right? Weeds count. If the weeds have flowers, all you need is at least 1 flower. So often, if it's an area that hasn't been mowed for a while, you can use clover, you can use dandelions. So often those might not be the first things that students think of, but they will work perfectly fine for this lab activity. So when they're going to be doing this, we're going to ask them some questions about the percent cover that's in different kinds of vegetation, like what percentage of this quadrat is in flowers versus not in flowers, or what percentage is in bare ground. That will also be part of the training activity. And it's not going to be an exact percentage between 1 and 100. It's a qualitative score, right? Is this less than 25? Is this more than 50%? So we can help students answer that question by using some training materials and background. One of the other things to consider, especially when we're looking for beetle pollinators, is you don't have to do this on the ground. One of the things I want to highlight, especially for anyone who's, let's say, using a wheelchair or mobility device, is you do not have to do this kneeling on the ground or sitting on the ground. You can use a tree or a shrub or an elevated container garden to do this lab activity. So we can make sure this is really scalable to people with all different kinds of mobility needs and whatever is around them. So this tall bush here, right, we can also do our observations on there. You can use plants that are ornamental and have been planted. So the point though is you have to find at least 1 flower. It can be in a container, it can be a tree. Some of the things that are actually out now with these larger flowers like these are actually ones that are really great for beetles. They like anything that's very smelly, for example. We'll talk more about thinking like a beetle in the landscape. They really like. So there's going to be some questions about the site in terms of the context. Is it urban? Is it rural? Is it a suburban setting? Are they in a park? Now, one of the goals in terms of making sure if they don't have backyard space that they can do this safely and legally is that they're not going to be doing any kind of destructive sampling, right? So if you place this quadrat and then you remove it after, we're not going to be in this particular core module destroying any plants. We're not going to be collecting any specimens. You don't have to worry about permits in that sense, right? So we don't have to worry that if they want to go do this in a state park, that they need a special permit to do that either. Okay, so then they're going to be taking a 10-minute observation, and they're going to write down some information about the kind of flower that we see. When we talk more about elements that are a little bit tricky and are in the guide, we'll explain what counts as a flower is important here. But they're basically going to be using either a paper data sheet or a like a Google form on their phone, whichever works better for your class, to enter data about tallying up what they see here. They are not recording insects to species. There are 9 functional groupings of pollinator, basically really big groupings, like, is this a bumblebee? Is this a beetle? Is this a wasp? Is this a honeybee? That level of organization. And so those are functional groupings that are actually standard in the literature. So don't think that just because we're not having them identified as species that it's not legit research, right? So these are functional groupings that are standard in the pollinating insect literature. Okay, later then, once they've done this, we want to compile our data together. This is actually data from some students who are working with me this summer doing the same exact time focal floral observations, just in some different habitats. One of the things I want to point out to you here is that for several of these, right, you can see they entered how many flowers, what kind of flower shape, we'll talk more about that, if it was more or less than 0.5% cover in flowers, whether or not it was really windy or no wind, some sky conditions. Then if you actually look farther to the right about what kinds of data they found, often they found nothing. So sometimes you can sit there for 10 minutes and find absolutely nothing. It doesn't mean it didn't work. It means that that's a true 0. And that's one of the things we can also help highlight to students. So one of the things though that I will do, I plan on video recording a 10-minute quadrat. If you have students, for example, who are quarantining and they can't go out and do this activity, we want them to be able to be involved in the lab and get that experience of looking at a quadrat. But we'll also pre-provide some data that's from other places that, like this, that really is other students going out and doing these observations, so that if they can't collect the data themselves firsthand, they can still participate in the activity using real data. Okay, so they're going to compile the data, they're from their own site and then from their class, and then we want to be able to look at patterns. So there's a bunch of different ways we can do this based on how quantitative your class is. So here's an example of what you can literally hand draw. You can hand draw an interaction plot like this, where every single, those are green dots, for example, are the plants, and the insects, in our case, it wouldn't be species, it would be functional groups, are the yellow dots. And you literally draw the interactions between them in the web. So you can do this on the scale of my individual observation or all of the class's observations. These are called bipartite plots, and you can also code them in R. So if your class is one that is comfortable in R and is already using them, you can actually just run the code and make these. But if your class is not comfortable doing that, you can also hand draw these. So it's very scalable in the sense of what you're hoping to see. You can also plot other kinds of information that's more exploratory with your data. You don't necessarily need to make bipartite plots. Okay, we'll then have the ability to compare results with other classes, with other studies. For example, this was that same paper that was done in France and published in 2013, and compare the results with our hypotheses. So one of the goals, right, is to make sure that students aren't just involved in the data collection, but see every part of the research process and participate in it. And so that's the goal. So we're going to give them other data sets to compare their results to, and then make conclusions about their data sets, and then in dialogue with other research. Okay. So now I want to walk through the 4 or 5 kind of add-ons that are possible. That's the core, and that's where collectively we'll all be sharing the data set together. But there's lots of other cool things that we can do with just a little bit more time. So one is to really explain to students phenomenology, getting into the mindset of your study organism. This is critical for natural history. This is critical to do organismal biology or behavior, if you're someone who studies animal behavior, you need to actually think like your study organism. So how can we help students do this? We're going to ask them to actually make a qualitative participatory map around their quadrat, so not of the quadrat itself, but of the area, and they will not be looking at it as a person, they'll be looking at it as a bee or a beetle, right? And that can also be multisensory, right? What am I smelling in the landscape? What am I hearing in the landscape? How would I represent that here? So in part is to get them to understand the value of qualitative data in the landscape, but also when they look at this picture, for example, do they see a building and a walkway? Those are things that people would be using, but how do we make sense of this landscape in a way that a bee or a beetle would? And then we'll also use that with a different set of reflection questions to ask what makes habitat good or better or worse for insect pollinators, right? So we don't want them to see a landscape and say, well, this one's better for me. What are the things in it they can reflect on in terms of what would make habitat better or worse quality for beetles or other insect pollinators? So if you chose to add that, there would be some additional background that could be a video or a PowerPoint slides or some reading, an additional data sheet of them recording that qualitative information while out at their plot. They could compile that information collectively as a class and look at what elements of those sites who had better and worse habitat in terms of when they're thinking like a bee or a beetle. And then some different questions they can ask themselves and each other about those sites using that addition of qualitative research. So the additional learning goals are to apply those concepts and then to understand the value of qualitative environmental data here. And then this would incorporate NEON elements that are about how people set up studies, right? So often we incorporate this as biologists or community scientists. We don't always make it explicit to students, right? So if you want to study a snake, for example, how do you know where you're going to find the snake, right? This is often implicit, and we want to make it explicit so we can train students in this. Okay. Another one, and this is an add-on module because it may not be possible for everyone's class or every student depending on where they live, is to actually do some pitfall trapping. So for example, if I want to actually go out and do an observation of plant-insect interactions in my backyard, what I would do after if I was going to do this is actually set up a pitfall trap and leave it, for example, for a few hours during the day or leave it overnight and come back to it. So if you've never set up a pitfall trap, it can be as simple as actually just digging a plastic cup into your yard, right, and leaving it. This is actually a picture from NEON because they cover theirs, but we're able to actually use in practice the same kind of method that NEON itself is using. NEON identifies all of their ground beetles that they sample to species. So we can do this activity, and you can either choose to release all of your study organisms. You don't need to necessarily put anything toxic in those cups. We don't necessarily need to do a lethal take on these organisms. It can be another way to do an observation, but students may not be in a place where they're allowed to dig holes and leave equipment. So this might be a great thing that works for someone's class, but it might not be a possibility for others. So if you chose to incorporate this, there would be additional materials needed, basically another training about how to actually set up the pitfall trap and collect it, including some information about how to not ideally get bycatch. I've accidentally killed things that we didn't intend to kill by leaving out a pitfall trap. So if they were to do this too, we'd want to be able to record it and find out what they're finding, compile it as a class, analyze that ground beetle data, and ask some other questions. Now there's a couple things I want to mention. The learning goals for this would be to actually perform the activity. So they would learn how to set a pitfall, learn why people do this, and then actually try it out themselves. And they would understand the value of either catch and release or lethal collection techniques for long-term biodiversity research, right? So observation is not the only method that people use to do long-term standardized research. This would directly use and incorporate NEON ground beetle sampling methods, but the caveat I want to say here is that NEON specifically identifies one family of beetle, like Carabidae. Many pollinating beetles are in other families. Many of the pollinating beetles tend to be more flying beetles. We may not catch any of them in a pitfall trap. So this is science, right? It's an experiment. We don't know that there will be overlap there. So for that reason in particular, anyone who uses this add-on for the module would also be using an additional hypothesis test, which would be looking and building off of the ground beetle literature to look at urbanization in the landscape and ground beetles in particular. Because we don't know that setting out a pitfall that we would actually catch any of the same beetles that would be out. Now, many of them walk on the ground, but I'm not sure, they might fly out of the trap in practice. Okay. So another one, if they do the pitfall trap, One thing they can do at home, again, depending on the goals of your class, is actually pin one of their specimens. So this is something that could be very useful if you're doing this, for example, for an intro or non-majors entomology class, but it may not be something that you want to do if this isn't one of the goals already for your class. And they would learn how to pin their insect at home. So the goal would be ideally to do this for one of the beetles that was caught in their pitfall trap if they do that activity. And the value of doing this in part is, again, to be linked up with the NEON data. The 2 maps on the bottom here are showing the sites where they do pitfall trapping of ground beetles at all the NEON sites. And that top picture is just a NEON photo of some of the pinned beetles in their biorepository. So the goal of doing this in part is to help students walk through that process if they're learning how to pin insects. And so they would need to also learn some more information about how to pin an insect. They would need materials in order to pin that insect. They would actually have to pin the insect, and they would answer some other questions about that. So there would be other materials and time needed if they wanted to do this. Okay, so the additional learning goals for this one would be to perform the technique appropriately with an insect from the trap and to understand the value of museum specimens. So that would be information that comes from the additional background material. Why do people use collections in long-term biodiversity research? All right, and that would use the NEON Biorepository data portal and some of the data from the portal itself if they want to do that. So they could actually compare what they find with what NEON finds in their bioregion. All right, another is to look at phenology in particular. So this is an example just from the PhenoCam gallery, but these digital cameras that are taking repeat photography to look at changes in the landscape. This is a great way to explain to students and tie in that when we think about habitat quality, we're not just interested in space. Habitat changes over time. So for example, what's flowering changes over time. And so we can actually incorporate and think about NEON canopy data and all the images that are available in the open access PhenoCam gallery to ask students to think about change over time, even within a season. So this, for example, is a rice field. We might be looking at this saying this is not necessarily great habitat for the things that we want to study in a floral observation. Most of the gallery pictures itself will not be things where they're not going to be at the scale where they could actually see an insect in these pictures, but they're going to learn about this technique. And what they can do then is actually be encouraged to take observations multiple times in their semester. So if you want to do this in your own experiment, what you'd basically be asking them to do is to do the lab activity of the core module at least twice. And if we do that, we would ask them to take a picture or to make note of different elements about their site, and then to think about what's changed over time between those 2 observations, right? What's flowering now that wasn't before? What insects are here that weren't before? To think about seasonality in terms of their sampling, right? It's a great thing for them to learn about. And so we can incorporate the PhenoCam imagery and explain why NEON uses it, even though it's not exactly the same. So if you want to add this on, there'd be additional background about repeat photography as a method, why people use it historically, why NEON in particular uses it with PhenoCam. They would be collecting additional activity and essentially repeating the core module at least one more time, which would mean they would also have more data to enter and compare with each other. They would have to analyze some different questions about change over time. So they would answer a couple of other reflection questions. So if it's a possibility for your group to do this twice in the semester, that could be a good fit. All right, and so the additional learning goals would be to identify and give examples of how habitat quality can change over time, not just in space. And it's, again, a qualitative measure of environmental data they could use. And we would incorporate the NEON protocol and data of PhenoCam. All right, the last one, I promise, is then to look at trophic interactions. So this might be a good one for some classes and not others. And so we want to think about how the image is not showing up for you, but beetles and other insect pollinators or other insects in general are shaped by both bottom up and top down forces. So this would be to help students explore that. They would not be collecting additional information. So for example, we didn't want to say you have to collect soil samples and analyze them because many students wouldn't necessarily have the ability to do that at home with the materials they have on site. But we could ask them to learn about that through some additional background material, and they could actually analyze NEON data, looking at soil characteristics, plant abundance and diversity information, and ground beetle data from different sites and over time. So they would be analyzing existing NEON data as the add-on, not additional data that they collected. And they would answer some other reflection questions about that activity. Okay, so the goal here would be to relate soil and vegetation, to the insect and in particular ground beetles between different sites that are at NEON sites. All right, what else though is here that might be useful depending on what your class is thinking about? I've already created an iNaturalist project space. Some people are really excited about uploading and they want to know what species it is. So there is an iNaturalist project space that's optional. So some people are excited about that. You can definitely upload observations there. If a class then especially actually wants to identify plants and insects further and do species level identification, there are tools like iSeek that are incorporated with iNaturalist that could be really helpful. The others that I've made a folder of open access literature that could be assigned as either background reading or as an additional reading, let's say for someone to lead discussion, especially if you have something like an upper level class or you're thinking about asking students to read a paper and compare their results with. So this is a folder of materials that we can openly share and are not in violation of anyone's copyright. There are some really cool things in there. One of the things I want to point out as a lover of history is that people have been observing beetles on flowers for literally centuries. So 2 of the papers in that folder are from the 1890s, and there are people talking about in the published literature their observations of floral observations. They weren't using a quadrat then, but it's a great way to help students understand that they're in a long line of people just paying really close attention to who's visiting flowers around them. So that kind of information might be a good thing that you want to assign if you want your students to be in dialogue with primary literature that's available to freely share with them, especially in a remote class. Okay, tricky things. Now, these are things that I want to say are not unsolvable, but they're things to be aware of, right? So that's one of the things that Lori and Tim asked us to do is think about what challenges might we run into while doing this. So there's a couple things that I think of, one of which that I think is important, and this is why there's an ID guide for both flowers and insects for this activity, is that what counts on a flower in the research litter for doing these focal floral observations depends on the type of flower. So an umbel counts as 1 flower, whereas a spike counts as 1 flower, whereas an open flower counts as 1 flower. So visits within the umbel do not count as multiple visits. Students will very much overcount. So for example, I want to show you in this next picture. These are 2 pictures from my yard, right? So that picture on the left, you can see that there are, on that evening primrose, there are 3 different kinds of floral spikes. So the whole spike would have to count as a flower, whereas this sunflower here counts as 1 flower. So it's not about size, it has to do with the kind of floral type. So that's something to just be aware of, that's what we need to orient students to as they would be doing this lab activity, would be to understand that what counts as a flower might be different than what they had learned about flowers as in a different context potentially. Okay, another is that in these counts, if this picture of this grass here was in my quadrat, this would not count. You're only counting insects that are doing a floral visit. If they're not on a flower or they don't land, it doesn't count. So this can be helpful for students to know, because sometimes if they see something flying by, they can be like, well, there's no way I could identify something that quickly. It's flying all around. If it's flying and it hasn't landed, they don't have to even try to tally it and observe it. So the great news to remind them of is that often pollinating insects are so focused on getting nectar or pollen, they want nothing to do with you, and you can often get very close to them and they will stay there for a little bit of time. So insects that are flitting around and don't land don't count. And if it's not on a flower, it doesn't count. All right, florivores, these are complicated. So not all beetles visiting flowers are necessarily pollinators, some of them are pests. So there are beetles, especially that are out now, that actually eat parts of flowers. They may inadvertently move pollen between a flower, but often they are not hairy, and so they're not doing that. So I wanted to show you some pictures that I took this past week where the beetles that I observed are considered pests, but they are actually on flowers. So these 2 were taken in the last week. That right-hand picture is a Japanese beetle that's actually feeding on clover. That was on my campus. And this picture on the left here is a rootworm that's actually feeding on sunflower in my yard. So these are beetles on flowers. And so I will include in the ID guide the kinds of fall flowers that, especially in temperate regions, we anticipate finding beetles on, and a list of plants that are more likely to have beetles in terms of flower color and size. So if they're interested in trying to say, well, I want to see a beetle, where might I go look? But just to be aware of, one of the reasons we want to talk sometimes about floral visitation and not just assume pollinator is that the activity that they're doing may not be pollination, even if they're definitely visiting a flower. But the way the technique works often when we're doing time focal floral observations is we're counting everything that's visiting a flower. But that's one of the reasons that we're not counting every particular kind of insect. We're often not counting things that we know would not be pollinators at all. Okay, so those are things to be aware of. The other things I had in here were mostly about disentangling different kinds of insects, but I know that we're going to run short on time, so I will pause for now and not run through this, depending on what the questions are. So Lori, I'll pause for now. I know you've been monitoring the chat. What kinds of questions are we?
Yeah, I did notice one that I think would be a good, it's actually the perfect moment. One person is asking, for the core data collection, should quadrats have only one type of flower, or are plots with multiple flowering plant species okay?
In an ideal world, we would want multiple kinds of plants, but I want everyone to know that you don't need to find that. So the minimum is literally one flower of any kind. If there are multiple kinds of flower, you will be able to build more complicated interaction networks from an individual plot.
Another question that just came up is, are we going to try to distinguish as we report the data ornamental and non-native flowers from native flowers?
Yes, in a qualitative sense. So students will not be asked to identify plants to species. So most of what the information we're going to have is going to be about, is this a cultivated garden? Is this an ornamental planting? If students are doing this on a campus or in a park, they may not have the answers to that information. So we're going to get some qualitative information to the best of our ability. It's one of the reasons people may want to upload pictures of flowers and get help doing that. One of the challenges of making sure this is scalable for everyone is not assuming that we know exactly what everyone has around them. And especially if students are doing this in their home environments, there are more ornamental horticultural varieties that would be less likely to be on the sort of like, these are the native wildflowers you might see around you lists. So one of the goals of doing this sort of as a pilot year is to test that out and see, you know, if we ask questions about cultivated versus garden setting, how deep can we get this? How close can we get this even taxonomically? Could we get most students study these to family? If not, what are the other things that we can identify that are functional?
Along those same lines, someone has raised that the most common and conspicuous fall beetle pollinator in their area is soldier beetles.
Mm-hmm.
And they're suggesting, what about focusing on a particular focal group for cross-site comparisons along with the broader basic protocol?
So you're saying, I just want to make sure I understand the question because I can't see it right now. Purposely looking for soldier beetles?
Right, or the idea is maybe a way to be consistent in the comparisons across sites are to only compare within a particular group.
Yeah, I mean, so the challenges of doing that at scale are that not everyone might have access to the flowers that soldier beetles would be on. There are also other families of pollinating beetles. So I think that that's definitely an option, would be to adapt this and subset it where your class is only looking for that, right? So you're not limited to exactly the protocol here. One of the goals though is if we do this in the way the core module is set up, we will all have the information about the morphospecies of pollinating insects collectively. So there's nothing limiting anyone from identifying beetles further. It's just that we also want to get that information about the functional groupings as a whole.
Another question is, I assume the protocol will tell me, but how do you handle site selection? Do you include a discussion of random sampling and biases in the protocol, I assume?
Yeah, this is tricky. I think what we have to do is basically raise the issue, but students are going to be limited if they're doing this off-site in where they can find flowers. So I think all we can do is encourage them to find flowers and to not pick poor quality habitat. one of the values of doing large scale observational field work, which is all the kind of field work that I do because it's in anthropogenic habitats, is we account for this kind of thing on the back end. Same with the training information. You can actually use the training quiz almost like the eBird people do, right? And you can link someone's training score with their observation. And if we need to account for that on the back end, we can. So I think we don't want to scare students off to say that if you don't have native wildflower habitat, you don't have legitimate flowers. One of the values of doing this is that we're doing it on what is out there and saying what is showing up, we want to know. It may mean that we get more zeros if it's a lot of low quality habitat, but these quadrats are very small and those are standards. So I don't think we're asking students to do random samples, and I think that that's okay.
This is a good time to ask this question. What about garden vegetables with flowers? Are those acceptable?
Definitely. So one of the questions about the site is this a garden? I don't think I have it in here, but I personally have been doing this in my garden, which is in my front yard and has sunflowers. So that's definitely an option. And it's also a great reminder to students, again, thinking about doing sites that are vertical, or if the flower is very large, that their quadrant doesn't have to be oriented down. It can be oriented sideways as long as they're looking at the same size area.
Let's see. There are a number of other people posting some interesting, experiences of their own. Maybe you have some insight into this one, Caitlin. Has anyone conducted such a project with beetles in an intro bio course for majors? In particular, students who may have not taken an ecology course yet, but are learning just the introductory principles of the field. And this person says, I think my intro bio students would be able to do this, but I'm curious about others' experience. So Caitlin, have you worked with very introductory students with this type of an exercise?
Yes, so I do this generally with a non-majors class, non-science majors. So they're not necessarily in biology at all. So that's one of the advantages of using a taxonomic grouping that is not at the species level. That said, I have not done it during COVID-19. So I think that one of the challenges is going to be figuring out, translating all of the training and figuring out how students are going to feel confident doing this on their own if they are literally on their own at home, is building that community because they won't have a lab partner out doing it with them.
Let's see. Kelly Lyons, who I know has worked with EREN before, brings up an important issue that we often run into. With EREN, I run into issues with biogeographic differences. If I ran this in the fall, a critical pollinator time in Texas, would that be still useful for the overall project? For example, if it were to go to publication?
Yeah, so that's one of the reasons that recording site and date information is really important. So one of the things I didn't yet mention, but I've talked with Lori about, is that we're not going to ask students, for example, to tell us where they live, but we need to have a nearby geographic point of reference that will help us make sense of this information. And then we will use the sampling date information to get a better sense of understanding that where people are in terms of the phenology of their area. So I don't see that as a barrier because, again, one of the basically groups that we're working with, right, with NEON data is we can get lots of cool insights at scale and recognize that there are these biogeographical differences, and it's not a problem, it's actually a richness to our data set together. So most of those issues, though, in terms of large observational data sets that are opportunistic, like all of us working together, but not on the same date, is that we just have to account for it when we're analyzing it in a research setting. So again, because we haven't done this project the previous year, this would be a new EREN project. We don't have an existing data set from last year that we can work through that with our students this year. We're gonna each be collecting in our own classes, and then basically from the winter forward, we'll have other people's data to be able to share and use to do it. But that's definitely not a problem.
Another question, does the protocol include taking plot and area photos? Actually, I guess you just addressed that, to facilitate site classification.
So I encourage, but not required. So again, I want this to be scalable for everyone's tech needs. We've heard mixed feedback of like, well, my students will definitely want to do this on their phone or want, links that they can do this on their phone. And other people are thinking, can I do this all with paper, or, you know, mail my students a lab packet and not expect access to the internet? So there will be both copies of the materials available for that reason. So if you want to include pictures, that would be great and be very interesting, but we don't want to make that a requirement because it would limit who else can participate.
So people have just made a number of very helpful observations about their own experience. Oh, there is one question about maybe using pan traps as opposed to pitfall traps. People were wondering if that would be an appropriate method.
I mean, definitely for pollinators, yes, pan traps. The only reason we are, the short answer is yes, we can definitely include an add-on about pan traps. I've done pan trapping. NEON does not do any pan trapping. So one of the things to just know about in terms of being in dialogue with NEON was that the only collection of terrestrial organisms that they're doing that includes beetles is ground beetles. So that was a NEON specific quirk about incorporating beetles and NEON data and this focus on land use gradients. But pan trapping is absolutely an option. I do think that would be lethal. So one thing to consider is just working through, you know, if you really want to do that and just what you're going to tell students about doing that and where it's okay for them to do that. So sometimes you can do a pitfall trap, and if you're using a smooth-sided cup, you can release everything the next day. You don't need to necessarily, you know, put soapy water or propylene glycol on the bottom, but a pan trap, you would be killing them. So just know that if you want to go that route. We were trying to avoid permits, avoid a lot of specialized equipment, but absolutely you could do panfall traps and see what you get.
Someone asks, and I think this is something that EREN can help coordinate with Caitlin as the leader. Will we have access to a list of participants and institutions as we collect, particularly with individuals in our region? I'm in the desert Southwest, and it'd be great to communicate with others. And I can just answer, as leader of EREN, we're very interested in facilitating those kinds of, you know, kind of regional networks or regional conversations and comparisons. So we'll definitely work with Caitlin to figure out how to, you know, kind of let people know who's part of the project. Mm-hmm. Often email lists kind of emerge from the project that are, that facilitate, you know, exchange, and it's very cool. There's also another question about emailing a link to the resources. My plan is to email everyone who registered for the webinar with information, but we're also going to be using the EREN website as a dispersal point. So the same place where you registered for the webinar, and I believe Tim has pasted a link again, yeah, into erenweb.org. That's a place that you can go and you know, get the materials once they're ready. So that should be cool. Let's see. Anyone else out there planning to work with pre-college students? Someone was asking. Yeah, please, you know, communicate with each other about this. This is cool. Let's see. I think we've gotten through most of the direct questions in the chat, and it is actually 4 o'clock. So I want to be very sensitive to people's time since we all are ridiculously busy at this particular moment in history and moment before the semester as well. But I thank everyone for joining us. Thank you very much to Caitlin. If you could please give her a virtual clap. She's doing a great job leading this project, and that is wonderful. And we are going to, of course, be having 3 more webinars this week. And those will be focused on plants across anthropogenic landscapes, on lichens, and on mosquitoes. So please join us. And thank you again to Caitlin. We'll see everyone later.
I hope to work with everybody on this. Yeah, bye.
you want to stop the recording, Lori?